Transformation efficiency. All right, this is a numerical measurement to determine the amount of genetically transformed E. coli cells. Basically, transformation efficiency is calculated to help scientists determine how well transformation is working. So basically, your transformation efficiency is expressed as the number of antibiotic-resistant colonies that also express your green fluorescent protein per microgram of plasmid DNA. So if you take a look at what we looked at, we are going to be concentrating on the transformation efficiency of our LB AMP arabinose sugar plate. Okay. Essentially, this transformation efficiency tells us the number of bacterial cells transformed by 1 microgram of DNA. The object is to determine the mass of the plasmid that was spread onto this LB ampicillin arabinose plate, and that was therefore responsible for the number of transformed colonies that you have observed that are fluorescent. In order to begin calculating the efficiency of your transformation, you will need 2 pieces of information. The first piece of information is pretty simple to obtain. You will need to know the number of green fluorescent colonies that are growing on your LB ampicillin arabinose plate. So what you guys are going to do ahead of time is you're going to count up all those colonies. The second piece of information that you're going to need is the mass or the total amount of the pGLO plasmid DNA in the bacterial cell that was spread onto that 4th plate, your LB ampicillin arabinose plate. Now this second piece of information requires a few more steps. You're going to need 2 pieces of information to find out that amount. Okay, you're going to need to calculate the total amount of DNA, which is what we used at the beginning of the experiment, and the fraction of DNA that actually got spread onto that 4th plate. All right, once you have calculated all of these numbers, you will then multiply that total amount of DNA used by the fraction of the DNA that you spread onto that 4th plate. This is going to tell you the amount of pGLO DNA in the bacterial cells that were spread. Okay, so let's do this second step out. First, you want to determine the total mass of the plasmid used. This is calculated by taking the concentration of DNA plasmid, which is in micrograms per microliter, and you're going to multiply that by the volume of plasmid in microliters. Alright, so for example, let's get rid of our picture here. And we're going to, you know ahead of time that you have 10 microliters of your pGLO plasmid. Okay? And you know also ahead of time that your concentration is 0.08, and this is going to be in micrograms per microliter. Alright. Basically, this means that for each microliter of solution, you have 0.08 micrograms of your pGLO DNA. So when you multiply that by your 10 microliters, you're going to be multiplying these guys together, you will get a total amount of DNA at 0.8 micrograms. Okay, this mass is important, and you're going to be using this again in your next step. Alright. Okay, so let me erase all of this. Alright, and then next what you need to do is you need to determine the fraction of pGLO plasmid of DNA that actually got spread onto that 4th plate. And by finding this, you need to first divide the volume of DNA spread onto the LB ampicillin plate, and then you're going to divide that by the total sample volume of liquid in the test tube. And that you're going to have to also calculate. So for example, you know that you spread 100 microliters, so I'm going to write that, 100 microliters, okay, were spread, and this was taken from a test tube. And this test tube, so let's pretend this is your test tube. Okay, and inside of this test tube, we added 250 microliters of luria broth. What else did we add? We also added, within this test tube, 250 microliters of your calcium chloride, which is also your transformation solution. So right now that's 500. Okay, it's all in there. And the last thing that was added, you can't forget, is your 10 microliters of your plasmid. Okay, so this gives you a total of 510 microliters. This is going to be your total volume. So what you're going to do is you're going to take 100 microliters and you're going to divide that by 510 microliters. These cancel out and you're left with 0.2. So that is your fraction of DNA. Okay. All right, now you want to know how many micrograms of your pGLO DNA that you spread onto that fourth plate. Basically, the total mass of plasmid in the fraction that you spread on that plate, because you didn't dump the entire container, you just dumped a fraction of it. So you're going to multiply the total amount of your pGLO DNA. So let me erase this. Alright, so we knew the mass from before, which was 0.8. So remember, this was the first step that I said for you guys to remember. So that is your mass of your pGLO DNA. Okay? And you also want to multiply that by the fraction that we just calculated, which was your 0.2. Remember, no units. Okay, so you're going to multiply those two together, and you're going to get 0.16 micrograms. So this is the mass that you put on that fourth plate. Okay. This is the end of your step 2. So we have this number here. So, in order to calculate out your transformation efficiency, okay, you're going to take the number of colonies that you counted, and you're going to divide that by the mass of the DNA spread on your agar plate. So, let's pretend that you had 190 colonies. And you, remember from before, calculated that you had a mass of 0.16 micrograms of your pGLO plasmid. Okay, this is going to give you a transformation efficiency of 1,187 transformed colonies per microgram of plasmid. And that is transformation efficiency.